Hi everyone, I am Jose Ordovas and I welcome all of you to this week's HNRCA Monday seminar. I am really excited to introduce today's speaker, Dr. Kelsey Mangano. Many of you know Kelsey as an outstanding and long-time collaborator and a familiar face in our community. She brings a ton of insight and energy to everything that she does. And today's talk, Unveiling the Diet-Bone Connection, Insights from Metabolomics and the Gut Microbiome, is not exception. Kelsey is an associate professor at UMass Lowell in the Department of Biomedical and Nutritional Sciences. She is also the director of the Undergraduate Nutrition Program and associate director of the Center for Population Health. And if that was not enough, she also holds an adjunct position at the Marcus Institute of Hebrew Senior Life, affiliated with Harvard Medical School. Her research focuses on how what we eat, especially things like dairy, fermented foods, and even food additives, influences our gut health, bone health, and overall aging. She's especially passionate about translating this work into real-world health improvements, particularly for older adults. Back in time, Kelsey earned her PhD in nutritional sciences from UConn and went on to complete postdoctoral training at the Harvard Medical Institute for Aging Research. Her work has been published widely, and we have the honor of co-publish with her, and recognized with several honors. But more than that, she is just a fantastic scientist, mentor, and communicator. A quick reminder before we start, feel free to post your questions in the Q&A box through the talk. We'll get to them during the discussion at the end. With that, please join me in welcoming Dr. Kelsey Mangano. Kelsey?
Wonderful. Thank you so much. Thank you, Dr. Ordovas, for that kind introduction. And it really, truly is an honor to be invited to speak here today at this prestigious research seminar and to have worked with so many colleagues at the HNRCA. I'm really thrilled to highlight some of the work that we've collaborated on during my talk this morning. So I'm excited to share some of this new work with you all. Starting off, just listing some of my disclosures that I thought were relevant to the topics that'll be discussed this afternoon, I guess. And my two major goals of this presentation is to outline major nutrients, food groups, and dietary patterns that are related to bone health. My lab has focused on nutrition as it relates to musculoskeletal aging for just over the past 15 years. And so I'll provide an updated overview and some existing gaps in the field as a whole, and highlight here and there where we've contributed to that area of science. And then the focus will be on the latter half of the presentation, where I'll describe some newer research studies where we're better understanding mechanisms underlying these associations between dietary intake and bone health, particularly in aging populations. And as I put these research presentations together, this one included, I thought to myself how over the past 15, 20 years, even when I started working in trying to reduce musculoskeletal aging, that I thought by now this first slide would be different about 20 years later. But unfortunately, it's not. And osteoporosis, or low bone mass, remains a major public health crisis. And this is one of the areas that my group continues to try to prevent. So 1 in 2 women and up to 1 in 4 men over the age of 50 will break a bone due to osteoporosis in their lifetime. And unfortunately, the burden of these osteoporotic fractures is great, both from a financial standpoint and also from a physical and mental capacity. So each year in the US, approximately 300,000 hip fractures occur. And about 25% of those hip fractures within a year result in mortality. And unfortunately, another 25% of those yearly osteoporotic related fractures end in individuals being bedridden in long-term care and assisted facilities. And of the remaining 50% of individuals that are able to walk and sustain independent living following a hip fracture, actually, surprisingly, only a small fraction are able to do this without an assistive walking device. So as you can see, the burden of osteoporotic fracture is still great, both in number and also for physical and personal and economic burden. The determinants of risk fracture are great, and my lab focuses on the nutrition-related factors that influence both mechanical overload and mechanical incompetence, which collectively lead to fracture. Nutrition can also be a major asset in the rehabilitation and prevention of additional fracture later in life, but the talk today will focus on nutrition's influence on the four major constituents that make up our mechanical competence or ability of our bones. The majority of work that has assessed diet as it relates to bone health or the prevention of low bone mass or osteoporosis with aging has really focused on this first asset, so bone mass, which is traditionally measured by DEXA. Newer research and a consensus has now helped us to realize that assessing bone mass is actually only a 60% explanatory factor of bone fragility, or why we may fracture due to osteoporosis. Another three components that are hypothesized to make up the other 40% risk are bone geometry, microstructure, and material level properties, or bone strength. Some of that is depicted here in the lower right-hand corner of the screen, healthy bone mass on the left and lower bone mass or osteoporotic bone on the right. And you'll see that the inner spongy tissue, the trabecular tissue, becomes more solid with osteoporotic tissue. There's fewer trabeculi, and it becomes less of a bone tissue that's able to exert and receive strength or force as you might wish, so that you can receive the force and not incur a fracture. And at the same time, with bone loss, you'll see that the outer layer or the cortical bone mass becomes quite thin and fragile as well, leading to higher risk of bone fracture. So these are two additional components to bone microstructure that additionally explain risk of fracture above and beyond bone mass. And so part of what my lab has moved into doing now, in conjunction with collaborators at the HNRCA, is to better understand how does diet influence these other components of mechanical competence? And if it does it affect them differentially? Or what are the hopes in prevention, not just of reducing loss of bone mass with age, but loss of geometry, microstructure, and strength? Nutrients can influence bone via multiple pathways. First and foremost, through proper nutrition, we're able to maintain health of our blood vessels and vasculature, which allows for greater delivery of nutrients to tissues and greater competence of those tissues to be able to take in nutrients and utilize them properly. Nutrition also aids in hormonal signaling, making sure that both men and women optimize the production of hormones that are anabolic for bone. Nutrition is known to directly reduce pro-inflammatory cytokines, and those cytokines have the direct ability to either suppress osteoporotic related bone cells, or with proper nutrition to allow for the formation of bone because of the suppression of these anti-inflammatory cytokines. Nutrition, depending on the type of nutrient that's consumed, is also utilized to build the matrix of bone directly. And other nutrients are anabolic for bone, meaning that they help to differentiate whether adipocytes, bone marrow cells, and hematopoietic cells differentiate into bone resorping cells or bone formation cells. So nutrition can influence bone via multiple pathways, and these pathways are often happening at the same time. So to get to sort of the root of what we're discussing today, what is nutrition and how can it influence bone? Is there an actual diet that we could define that would optimize our bone health or reduce the loss that we incur with age? And there are a lot of different groups that have made suggestions as to what a bone healthy diet could be. I've pulled in one example here from the European Society for Clinical and Economic Aspects of Osteoporosis and Osteoarthritis. And you'll see that their bone healthy diet focuses primarily on nutrients, protein, vitamin D, and calcium. But what's interesting is that they've now updated these guidelines to include a higher density of protein recommended, particularly for older adults to achieve optimal bone mass. And they also give more specific recommendations now in terms of amount per meal or bolus amount to optimize the formation of new bone. With vitamin D, the recommendations are generally the same in terms of meeting the DRI and meeting sufficiency levels in blood. And for calcium, what's interesting is not only alongside the DRI for adults or 1,200 milligrams per day for older adults, they also now interweave the necessity of regular physical activity alongside these dietary intakes to really optimize the use of nutrition in feeding our musculoskeletal system. And they also now briefly mention timing and how the timing of nutritional constituents could affect bone and muscle tissue differentially. So although the focus still remains on individual nutrients with official guidelines, they are starting to weave in some newer consensus of research that suggest timing and the interrelation of these nutrients and the interrelation with physical activity is equally as important. What still is being pulled together by scientists and collection of researchers to put forward to add to this bone healthy diet recommendation is continued investigation into whether the variety of nutrition intake matters, the nutrient density of those food products. Do individual foods matter than others, or is it the collection of foods within a food group? And how bioavailable are each of those sources? So there's additional metrics that could be layered into these recommendations moving forward. I've put together sort of a very simplistic, almost stick figure-like consensus related to information from over a handful of recent systemic and meta-analyses that have been published just within the last 5 years that really do a nice job summarizing overall nutritional components and food groups that may be either positive for bone, highlighted here in green, or have negative attributes towards bone, highlighted in red. And the thicker the line, the greater consensus of research, and a dotted line or a thinner line is attempting to indicate moderate level evidence versus high level evidence. And so that you'll see on top of traditional recommendations for calcium and vitamin D, other nutrients have not only gained interest over the past couple decades, but there has been a consensus of research surrounding a lot of these, not only nutrients, but now food groups that could influence bone health in a positive way and be considered into what we recommend for patients to prevent bone loss with age. You'll see that I do have some question marks that are still here because we are still trying to disentangle, again, whether the type of food matters within food groupings. Does fat content matter for dairy foods? Does the type of dairy food matter because they provide different nutrients? Not all dairy foods are equal. The same goes for fruit and vegetable intakes. So there's a level of research relating, and I think this isn't for all fields, not just with bone, where we're looking to go back down into individual foods for personalized nutrition, but then we still have this need to expand to look at overall dietary patterns in terms of, will people eat foods and they eat them in groupings, and how can we give broad stroke recommendations to the average American without nutrition knowledge to prevent loss in bone. So there still seems to be this balance in continuing to assess individual food groups and whether or not personalization can help certain groups of people, and also looking at overall dietary patterns and if we can define this bone healthy diet. The majority of work out of my group is continuing to look at fruit and vegetable intakes, and that's what I'll be discussing today, and also dairy foods and overall dietary patterns that actually capture all of the components that are listed here on the screen. Some of the work that we did in terms of how does total protein impact bone health, and do dairy foods play a particular role in this, and do processed foods matter? One of the ways that we can assess dietary intake and overall patterning and how it relates to disease outcome is to not only consider how foods are consumed within the context of total calorie intake, but for individual macronutrients, does food contribution, in this example, to total protein intake matter? So is it beyond total protein accruement, day in and day out, grams per day, or does food source matter? As has been indicated, particularly for older adults, it seems that food source plays more of a role as we age due to the blunting of amino acid receipt at the muscle and bone tissue. And so our work in the oldest generation of the Framingham cohort did show that there were differences between the protein-specific food clusters that we created, particularly at the femoral neck, where the majority of protein that was coming from low-fat dairy, which was driven in this group by majority of milk intake, had significantly higher femoral neck BMD compared to individuals where highest protein intake came from either red meat or processed protein foods. And although not statistically significant across any of the other sites, you can see a similar pattern was observed in differences between these protein-driven food groups and its relation to bone mineral density. Many other groups have looked at dietary patterning, either a priori patterns or posteriori, and this is a meta-analysis of posteriori dietary patterns across, I believe it was 10 studies related to fracture risk and 20 studies related to bone mineral density. And on this slide, I'm showing you the relative risk according to either individuals following a healthy dietary pattern, which was largely driven by high fruit and vegetable intakes, whole grains, nuts and legumes, and dairy, versus individuals that more closely followed what they labeled as a meat or westernized dietary pattern with more of the processed foods and higher red meat consumption and lower fiber intake. And you'll see that across these 10 to 13 studies, a healthy dietary pattern was significantly related to lower risk of fracture in their lifetime versus individuals who followed closer to a westernized style diet. There was a slightly elevated risk for fracture in their lifetime. Again, trying to capture the totality of the diet and future risk of fracture. The same meta-analysis also evaluated whether dietary pattern was related to bone mineral density. When they looked at the 20 studies, not only did a healthy dietary pattern and a meat or Western pattern seem to be consistent across these 20 studies, but a dairy pattern also was independent this time from the healthy pattern when related to bone. And they again found that individuals following a healthy dietary pattern tended to show lower risk of fracture in their lifetime versus, again, individuals following the meat or Western dietary pattern with higher risk of fracture. And then the dairy group in itself, when it pulled out into its own pattern, also showed a significant reduction of risk fracture in lifetime. So indicating that we could potentially partition out or help individuals to understand a healthy dietary pattern, generally similar to the dietary guidelines for Americans, which do include high whole grains. Dairy intake is still included on my plate, and focusing on fruit and vegetable and lean protein consumption. Now, although these meta-analyses have been formed, it does seem to be a consensus of research surrounding healthy dietary patterns, optimized bone health on top of adequate calcium, protein, and vitamin D intake. There are remaining questions as to whether or not those recommendations for the average American could be further personalized to other subgroups that might experience osteoporosis earlier in life, may experience it at greater prevalence rates, and may also have other underlying health conditions that could exacerbate or alter dietary interactions with bone health. So some remaining questions remain in terms of differences between biological sex, race and ethnicity, genetic ancestry, and as I said, whether or not somebody has underlying health conditions. An example being dairy intake has generally been shown in the US to be advantageous towards bone when focused on lower fat options. Whereas a more recent consensus of information coming out of northern countries or Nordic countries in Europe have actually shown quite the opposite, that higher fat dairy products are showing a greater consensus against protection of bone versus lower fat components. And one of the hypotheses of why this would differ by population is that Americans have a greater level of metabolic disease. So in the context of metabolic disease, could dairy products based on fat component affect bone differently? Or within the context of the caloric intake, lower fat intake is more beneficial both for total body health and also for bones versus a healthier, lesser metabolically disturbed population in Northern European countries seeing benefits from fat in dairy. And so other underlying health conditions can certainly interact with how nutrition may impact bone in our lifetime. And so our group asked, could understanding these mechanisms better help us to understand whether or not personalization of dietary recommendations for reducing musculoskeletal losses could really benefit groups of people, particularly with high risk for bone. And so our research group, all of the data that I'll be showing you here on out is from the Boston Puerto Rican Health Study and the Boston Puerto Rican Osteoporosis Study, which is a longitudinal cohort of adults who self-identify as Puerto Rican who live in the greater Boston area. The study began in 2004 under the direction of Dr. Catherine Tucker. And she was able to recruit over 1,500 adults between the ages of 45 and 75 years of age and followed them for 13 years. We just finished our last follow-up visit in 2022, and we had around 500 participants either remaining in the area and were still alive and willing to participate. So the research I'll show you moving forward is from both of these cohorts. One of the reasons why we wanted to assess musculoskeletal loss with age and bone mass losses with age, essentially, in Puerto Rican adults was because of some of this data that was published by Ann Looker from NHANES data that was depicting prevalence rates of either osteoporosis or low bone mass across women and men. And we were interested in this light blue bar that showed interesting results to us that particularly in women with osteoporosis, Hispanic adults had the second highest prevalence rate of osteoporosis and the highest rate of low bone mass in women. And in men, the second highest rate of low bone mass and the second highest rate of osteoporosis compared to non-Hispanic Black, non-Hispanic Asian, and non-Hispanic white adults. And so Dr. Sabrina Noel and I published a paper specifically looking at risk of osteoporosis and prevalence of low bone mass in the Boston Puerto Rican Health Study. And this was a part of the initial bone wave conducted in the early 2000s. And what we showed was that this is age-adjusted percentage of osteoporosis in women on the top panel and men on the bottom panel. The black bars indicate osteoporosis and the gray bars represent percentage of individuals with low bone mass. And in women in the first column, Puerto Rican adults, again, had the second highest rate of osteoporosis, second to that of Mexican Americans. And interestingly, men of Puerto Rican descent had the highest rates of osteoporosis at 8.6% compared to Mexican American, non-Hispanic white, and non-Hispanic Black adults. So this was really jarring for us in that we were connected to a group and a community of adults with high prevalence rates of osteoporosis that could have very unique cultural and biologic needs, and that we were very interested in assessing further how could diet specifically be related to bone in these individuals. So again, this is just a picture depicting the multiple waves of follow-up from the original Boston Puerto Rican Health Study that was started in 2004. I've highlighted here in yellow where bone was collected because that will be the focus of today's talk. Baseline bone started in 2007, and follow-up bone we just finished collecting in just under 500 adults in 2022. And so the first study assessing some of the underlying mechanisms, or trying to piece apart the underlying mechanisms connecting dietary quality to multiple measures of bone health, was really spearheaded by my postdoctoral research fellow, Lisa Merrill, that you can see on the upper left-hand corner of the screen. And so I just wanted to highlight Lisa's amazing work that I'll be presenting today in this talk. And so Lisa wanted to know if dietary quality was associated to bone mass in this cohort, but not only bone mass. Could we capture some of those other microarchitecture and strength components of bone that have been traditionally under-researched in their relation to diet? So we included some of those measures, and she also wanted to know if gut microbial health was directly in this pathway. And so this is a cross-sectional analysis because we collected stool at one of the last waves of follow-up, and we have one assessment of gut microbiome in these participants. We also collected measures of bone within a year of collecting those fecal samples. And then to assess dietary quality or the exposure, Lisa took an average of dietary intake across multiple waves of the Boston Puerto Rican Health Study in attempt to even better capture usual intake from the repeated FFQs that we collected over the 13 years of follow-up. Some of the original work that Lisa built off of in order to determine what measure of dietary quality would she use is based off of a publication that Sabrina and I published in 2020, where Sabrina evaluated 3 different a priori dietary indices: DASH, which is shown on this screen, dietary approaches to stop hypertension. We also assessed the Mediterranean diet score and the alternative healthy index, assessing whether people were adherent generally to the dietary guidelines for Americans. And we related all 3 of those dietary indices to bone mineral density or bone mass. And I'm just showing you DASH because that was the only dietary indice that was significantly related to bone mass in this Boston Puerto Rican cohort, where you can see in the highest quintile of adherence to this diet, they had significantly higher bone mineral density at 2 different measures of the HIP compared to adults in the lowest quintile of adherence to the DASH diet. And this is why Lisa chose to use DASH as her dietary indice of choice moving forward. But what was missing from our original publication was whether dietary quality was not only associated with a measure of bone quantity, but is it also associated with measures of bone quality? Just by quick reminder, the DASH score, it really encompasses a lot of those bone healthy nutrients and food groups that I had alluded to earlier. And this is a depiction. The DASH score has an adherence range of 8 to 40, with 40 being highest adherence to this diet. And positive loadings come from greater intake of fruits, vegetables, whole grains, low-fat dairy, nuts, seeds, and legumes, whereas sodium, sugar-sweetened beverages, and red and processed meats load negatively on this dietary quality score. Again, really getting at the totality of that original figure that I highlighted of nutrients and food groups that have been shown across a consensus of research studies to be beneficial towards bone health. Now, in addition to measuring dietary quality as the exposure, we also looked at multiple measures of bone health as the outcome. And this was under the direction of our colleague at the HNRC. All of these measurements were obtained under the direction of Dr. Beth Dawson-Hughes, who we are so grateful, and her entire team who helped us to collect data on these 500 or so participants. So we did do a follow-up assessment of bone quantity, or BMD, assessed by DEXA. But what we also wanted to assess were two different measures of bone quality. So one of the measures that we were able to calculate actually from the original DEXA scan is trabecular bone score, which is an indicator that can be used to assess the microarchitecture of bone tissue. And you'll see here what the trabecular bone score adds on top of the bone density scan is this figure is highlighting for you that these two individuals, patient 1 and patient 2, have the same exact T-score, -2.2, as derived from the DEXA scan, which would indicate that they would have equivalent measures of bone health. However, when we layer on the trabecular bone score, which uses all of the individual pixels taken from the DEXA scan, and it looks at the heterogeneity or the homogeneity of those pixels, the more homogeneous the pixels are, the more normal or toward normal the trabecular tissue is in terms of healthfulness. The more heterogeneous the pixels load, or the higher the trabecular bone score, the more degraded the spinal tissue in this instance is, and has lesser quality trabecular, that inner spongy bone. And so these individuals, although they have the same bone mineral density, actually show on top the higher score for more homogeneous bone, and a lower score because of their heterogeneous lack of trabeculi. So same bone mineral density, but quite different levels of bone quality. So that was one of the reasons why we captured trabecular bone score in our group, to try to capture more information about bone. And then we also performed a measurement of bone strength using the osteoprobe device, which is shown here on your screen, where individuals were applied with a local anesthetic, and then this pen was essentially driven into the tibia. And it performed a very small microfracture in the bone tissue, and the greater the depth of the pen that was able to insert into the bone, the greater the BMSI, or indicating a lower bone strength. So these were the three measures of bone health that we captured in this population. Now, Lisa was interested in making the connection between dietary intake and bone, but hypothesized that the differences between those who adhered higher to the DASH diet versus lower would see higher and low bone scores due to, in part, changes in the gut microbiome, or I should say differences, excuse me, in the gut microbiome, because this is cross-sectional. And I know the audience is well versed in how the diet has been shown to modulate our gut microbiome by the provision of nutrients directly to our gut, which can be further fermented by the microbes. In someone with a healthy gut, it increases the healthfulness of this green colored cartoon, or the mucus lining of the intestinal cells, which promotes healthy absorption of nutrients, and in this case nutrients that would be beneficial for bone. It also drives down inflammatory cytokines and protects the intestinal barrier, which allows nutrients to influx in to circulation and to keeping waste products and any type of inflammatory products to be excreted in the stool. Now, these mechanisms of a healthy diet can affect other downstream processes that have been shown through a consensus of recent research, particularly in animal models, to affect the creation or the differentiation of both bone building cells and bone breakdown cells, directly having an indirect influence on bone health over time. And so some of these hypostulated mechanisms include driving down inflammatory cytokines, which would favor bone building, enhancing the intestinal integrity, which enhances immunoregulatory responses, which again enhances bone creation and lowers bone breakdown. And it also will sort of shift the creation of other anabolic proteins in the gut that can be absorbed into circulation, and again affect these same processes. So due to these mechanisms, that was the rationale behind investigating whether in our population this was one of the mediators in that association between diet and bone. So Lisa looked at, again, dietary quality assessed by DASH score and in multivariable regression, whether or not it was related to bone quality measured by BMSI and TBS, and then bone quantity by BMD, both at the femoral neck and the total hip. And you'll see here that I've highlighted on the right that greater adherence to the DASH dietary score was significantly related to higher bone mineral density at 2 areas of the hip. And this is confirmatory from what Sabrina had previously published in this same cohort, but about 8 years earlier. What we were surprised to see was that DASH score was not related to either measure of bone material strength nor TBS, or a measure of bone quality. Now, the next question that Lisa wanted to ask, with now a greater focus on bone mineral density as our outcome, which is due to time constraints, what I'll focus on here, since it was the only bone outcome related linearly to dietary exposure, was to say, whether or not diet was related to any markers of taxa in the gut microbiome or related pathways. And so this figure here shows differentially abundant taxa that were significantly related to greater adherence to the DASH score, highlighted with these orange bars going to the right. So greater log fold in these 4 taxa was seen with greater adherence to the DASH diet. And then you'll also notice that just 3 taxa are highlighted here in gray, and these had a greater log fold abundance under the presence of higher sodium intake in this population. The top bacteria, L. elegans, is a healthful microbe that produces short-chain fatty acids. So this was confirmatory to some other research that has suggested that a healthy diet helps to form short-chain fatty acids, which can be absorbed into circulation and have a positive influence on bone health. The Clostridia and Ruminococcus taxa are difficult to interpret, since these microbes can be either beneficial or detrimental to the host, depending on the environment and the specific species. And as you can see by their listed names, our sequencing steps did not provide information at the species level for these taxa. And this is not uncommon, even in metagenomic sequencing, as reference databases are still incomplete. Some bacteria have high genomic variability. Bacteria frequently exchange genetic material, which can complicate classification, and there are still limitations in classification algorithms used for metagenomics. So although we observed differences in these taxa, we were not able to label the specific species and whether or not it had specific downstream functions that might be related to bone health. So the next step was to see whether or not the 7 taxa that were significantly related to diet were also indeed related to measures of bone health. And in this instance, I'm just showing you the results for bone mineral density. And so what Lisa did was she performed machine learning algorithms using random forest plots to ask whether or not, with the inclusion of those 7 taxa, and also the inclusion of known bone covariates, right? Age, BMI, height, vitamin D, the inclusion of known covariates plus those 7 taxa, what were the top 15 determinants of bone mineral density? And that's what I'm showing here on this screen. So using the random forest plots and using an assessment method called Shapley values, we were able to prioritize the top 15 factors that explained bone mineral density with the least amount of error. So these highlighted for us the 15 most important factors related to BMD in this specific cohort. And you'll see that some of the larger the bar, the greater the Shapley value, which indicates greater importance in that model, or the least amount of error across this model. And BMI, height, age, sex, serum, hydroxyvitamin D, not surprising that they were some of the greatest determinants of BMD. We also included an inflammatory biomarker score, which was a standardized score of 4 circulating markers of inflammation. And then you'll see I highlighted in blue or black here the 3 of the 7 taxa that were significantly related to greater adherence to DASH were in the top 15 factors that explained bone mineral density in this cohort. Now, one of the limitations to showing these Shapley values is it doesn't indicate the directionality of association. So that was the next step. On this slide, I'm showing a B-Schwarm plot, which indicates the directionality of each of the 15 factors that explained most of the variation in bone mineral density. And so the 15 factors are described along the y-axis. On the bottom to the right of the 0 indicates a relation to higher bone mineral density, and to the left of the line indicates relation with lower bone mineral density. Now, we interpret a factor related to high or low abundance based on its color coordination. So if we, for example, look at one of the taxa that was associated with greater adherence to DASH, you'll notice to the right of the line, there's a thick clump of dark purple, meaning that individuals had low abundance of this taxa, and individuals with lower abundance of the taxa had higher bone mineral density. And you'll see as some of the color goes up into the shades of pink and yellow, those individuals with a higher abundance of this taxa had lower bone mineral density. So indicating higher adherence to DASH upregulates the abundance of this taxa in the gut, which is related to having lower bone mineral density. So each of these have to be interpreted very carefully within the context of how it's related to DASH, its abundance in the gut, and whether or not it relates to high or low bone mineral density. But interestingly, these taxa, the Lachnospira elegans, for example, higher abundance of that taxa in the gut, the one that makes short chain fatty acids, was related to having higher bone mineral density. And some of the unrelated species were more difficult to interpret. And so we did perform these machine learning plots on the other outcomes on the assessment of bone quality, even though they weren't related to diet. And so I'm just showing you here a summary. Instead of showing you all of those plots, I'm providing a summary of all Shapley values and bone outcomes. So as I showed you with DEXA or BMD, excuse me, the short chain fatty acid producer, L. elegans, was related to higher adherence to DASH and greater bone mineral density, as were those 2 other unclassified taxas. And generally, the pathways that were associated with bone mineral density were related to energy metabolism. For bone quality, taxa associated were characterized as short chain fatty acid producers as well. And some of them also had other immune functions. Pathways associated with bone strength, particularly focused on inflammatory functions, whereas pathways that were significantly related to trabecular bone score showed more of a mixture of energy metabolism and also some of those inflammatory processes. What was interesting is that across all bone outcomes, whether it be quality or quantity, that inflammation biomarker score and serum hydroxy vitamin D was always present in the top 15 factors that predicted bone. Those were the 2 most consistent factors across all 3 models. Now, just to summarize this study before I highlight one other study we've been working on recently in this area, DASH did predict bone mineral density, but it wasn't predictive of bone quality, keeping in mind that these were linear models, so multivariable linear models that were used to explain bone. And the reason why I bring this up is because we've recently looked at some machine learning nonlinear plots, and this is one for TBS where you'll see I've highlighted in green that DASH is related to TBS, but in a nonlinear fashion, also using these random forest plots where greater adherence to DASH was related to higher TBS score. So future studies should really consider and be careful in assessing both linear and nonlinear associations between diet and bone. So for time, I'm just going to move on because I do want to make sure I highlight one other study with the HNRCA colleagues highlighted in the upper right-hand corner of the screen. We worked together to assess whether fruit and vegetable intake was protective of bone. But in this case, we looked at metabolomics and whether or not that mediated the association between diet and bone. And we had about 600 adults from the Boston, Puerto Rican health study where we could make this assessment. So first, using multivariable regression, we did show that in our population of Puerto Rican adults, greater fruit and vegetable intake was significantly related to lower odds of osteoporosis. And in addition to overall intake, fruit and vegetable variety month in and month out was also significantly related to lower odds of osteoporosis, both in men and women. What was, I think, the most surprising to all of us is that when we started to look at fruit and vegetable metabolites and then the osteoporotic related metabolites was that this consistently differed by sex. And so for us, this is one of the most important takeaway messages that when assessing diet and how it relates to bone, sex differences really has to be considered, particularly here for the metabolome. So we looked to see which whole body metabolites measured in blood were related to fruit and vegetable intakes. 66 metabolites were significantly related to fruit and vegetable intakes in women, where 38 metabolites were related in men. Interestingly, only 7 of these roughly, I think, 104 metabolites were shared between men and women, and I've highlighted those shared metabolites here. The beta coefficient indicating whether it was positively related to greater fruit and vegetable intake or inversely related. Only one of the metabolites, homoarginine, showed opposing directionality between men and women. All of the others showed shared or the same directionality with fruit and vegetable intake across both men and women, although the majority of the metabolites differed. And this is just showing one of the numerous volcano plots that we published where women are triangles and men are circles. And the color indicates individual metabolites within this metabolite group, the carnitine fatty acid group, that were significantly related to greater fruit and vegetable intake. They had a greater presence of these metabolites in their blood with higher fruit and vegetable intake. The next sequence of this study was to ask whether there were individual metabolites related to prevalence of osteoporosis. And again, we did find 33 metabolites that were related to lower odds of osteoporosis in women and 40 separate metabolites in men. Generally in women, the metabolites that were related to lower prevalence of osteoporosis were part of the steroid hormone biosynthesis pathway, and in men, the branched chain amino acid metabolite pathway. I think 10 of the 40 metabolites were from this pathway. Interestingly, of these metabolites related to presence or absence of osteoporosis, only 2 overlapped between the sexes. And you'll see that the top metabolite was higher with the presence of disease, both in men and women, but the second metabolite was lower in women with osteoporosis and higher in men with osteoporosis, again suggesting sex differences, not only in the identification of the metabolites, but potentially the directionality with the outcome. I do want to highlight that the metabolites related to presence of osteoporosis in our cohort were consistent with 3 other studies that had come out from the Twins UK cohort, Hong Kong cohort, and I'm sorry, I'm forgetting one other cohort that we compared to. These were all in women, but that the highlighted metabolites were consistent in that they showed these metabolites were related to bone, as did we in our Puerto Rican cohort. And then lastly, I'm just going to end by showing you that we performed PCA analysis of the fruit and vegetable intake. We showed 6 different identified groups of fruits and vegetables that were related with metabolites, different metabolites in women, again, than were observed in men, but I've highlighted berries and melons and dark leafy greens because those were the 2 food groups that consistently across both sexes showed that they either inhibited or enhanced metabolites that were directly related to lower prevalence of osteoporosis, suggesting that not only could total fruit and vegetable intake be beneficial, but helping to hypothesize for future intervention studies that specifically these two food groups could be of particular interest in preventing osteoporosis later in life. And so I do want to leave some time for questions, and I want to thank my collaborators for supporting these projects, and I look forward to answering any questions that you have. Thank you so much.
Thank you, Kelsey. Great talk. And yes, you have some questions waiting for you. The first one comes from Alice Lichtenstein. Thank you for a very interesting presentation. For the differences you observe between different sources of dietary protein and BMD, how closely are those differences related to amino acid scores for each protein type?
Oh, that's a great question. That's a great question, and we do have individual amino acid types quantified in both Framingham and the Boston Puerto Rican cohort, but I have to say that we didn't specifically look at amino acid profiles, which would, I think, directly get to your question and would be interesting. But even before going back and doing any replicate analyses, it would be interesting just to see across those food groups the similarity or dissimilarity of the amino acids provided by those foods. So long story short, I don't know the direct answer, but I really appreciate the question, and it's something that I can certainly look into because the question remains, to Dr. Lichtenstein's point, is it differences in amino acid composition, or could it also be differences in probiotic fermentation versus milk polar lipids versus fat content? And so I think it would be really interesting to dive into that amino acid fraction more specifically. So thank you for that question.
Thank you. The next question comes from Larissa Konstantinov. What is known about the role of vitamin K for bone health? Is there a benefit of combining vitamin D and vitamin K in dietary supplements? Can gut microbiome influence bone health via modulating of vitamin K synthesis?
Oh, those are great questions. I just wrote them down to make sure I can answer all of them. So to answer the first question, there is a strong consensus of research originating in mouse models, but now it has been replicated in a handful of human studies showing that vitamin K does positively influence bone mass. One of the ways is it helps to enhance the proliferation of osteoclast, the proliferation of that cell type. So vitamin K is known to improve bone health, and that is one of the reasons we hypostulated in our fruit and vegetable study, those metabolites that were upregulated by greater intake of the green leafy vegetables. Vitamin K is most densely found typically in those green leafy vegetables. So some of those metabolites were related to vitamin K metabolism. And I think that answers your third question. That certainly, I didn't show it here, but I think we can hypostulate and test in a future study that perhaps the gut microbiome could be acting on bone by enhancing vitamin K production in the gut. And also with higher green leafy vegetable intake, the provision of vitamin K itself would also enhance that. The second question on the interaction between vitamin D and vitamin K, that's a little bit outside of my wheelhouse, and I don't know of that interaction specifically with supplements, but I know through food, it seems to be a positive entity.
Thank you. And well, this will have to be the last question, the next question, because Kelsey has to continue teaching in the next few minutes. So this one from Shandong Wang, great talk, thanks. You mentioned that several specific gut bacteria, which are known for producing short-chain fatty acids, were associated with bone density in the Puerto Rican study, or the Puerto Rico study. I was wondering if you have examined plasma concentrations of short-chain fatty acids or their metabolomics, or your metabolomics, if in your study, and see any association to bone density.
That is a great question. No, we have not looked at plasma levels of short chain fatty acids, but to your point, Jose, I don't see why we couldn't check the metabolite list to see if some of them are derivatives of circulating short chain fatty acids. So we haven't looked at it, but I really appreciate the question because that is certainly something that we should do.
Yeah, and it's one of those things that, of course, is not clear in the literature because you see arrows pointing in all directions.
True.
And with this, many thanks again for your wonderful talk. I wish I could have listened to you 20 or 30 years ago
Okay.
and learn from what you just told us. And I think the current arrow points to next door, where you have to continue your teaching duties, right?
Exactly, yes. On to the next, but I really appreciate you inviting me and thank you everybody for your wonderful questions. I really appreciate it. And please contact me if you have other ideas or would like to collaborate. I'd love to collaborate, so please reach out.
Okay, and there are other questions that were not answered, so maybe what I can do is just shoot an email to you, and then you can answer them at your pleasure.
That would be wonderful. Thank you so
Thank you
much.
so much. Bye-bye, Kelsey.
Bye-bye.